Assalamualaikum and hello. We are from Group 5 and today we are going to present about Commercial Diagnostic Kits for Periodontal Disease. Periodontitis is a chronic inflammatory disease results in progressive destruction of the periodontium that supports the teeth and is of bacterial origin. The traditional methods for diagnosing periodontal disease have remained virtually unchanged and relies almost exclusively on clinical parameters such as probing depth and clinical attachment level, and traditional conventional dental radiography, particularly intraoral periapical radiograph and orthopantamogram. The strength of these traditional tools are their ease of use, their cost effectiveness, and that they are comparatively non-invasive. However, these diagnostic procedures are limited in that only disease history can be assessed. However, we need to develop newer diagnostic tests which can detect the presence of active disease as well as predict future disease progression and evaluate the response to periodontal therapy. Use of biomarker-based disease diagnostic that focus on the identification of active periodontal disease using gingival crevicular fluid, GCF, and saliva. Elevated level of these periodontopathogens may be useful indicators of both active periodontitis and increased risk of attachment loss. The active disease is diagnosed, the less invasive, the less time-consuming, and therefore less costly the required treatment can be achieved, and the better long-term prognosis for patients with the destructive disease.
I will talk about the characteristics of an ideal diagnostic test kit for periodontal disease. First, it should be quantitative, which means it can be measured. Second, it should be highly sensitive method capable of analyzing a single periodontal site in health as well as disease. Third, it should be reproducible. Fourth, highly specific. Fifth, simple to perform. Sixth, it should be a rapid procedure, which means it only takes about 1 or 2 stages procedure. 7. It should be non-invasive, which means it is safe to treat patient. 8. Versatile in terms of sample handling, storage, and transport. 9. Amendable to chairside use. 10. Economical. And lastly, dependent on simple and robust instrumentation.
Let's continue with microbiology diagnostic test kits available in markets. So we have Evaluate, Perioscan, Omnigene, and IAI Padu test. So for IAI Padu test, this test use oligonucleotide probes complementary to conserve fragments of the 16S RNA that encodes the rRNA which forms a subunit of the bacterial ribosome. The detection threshold of this test is 103 for AA, 104 for PG, T. forrestia, and Treponema denticola. And the disadvantages is this test seems to underestimate the number of positive sites or individuals suggested by a high number of false negatives.
The next diagnostic test kit will be Evolocite. It is a polyclonal antibody-based sandwich enzyme immunoassay. It involves linkage between antigen and a membrane-bound antibody to form immunocomplex that is revealed through the calorimetric reaction. Patient plaque samples are prepared by the addition of detergent and mixed together, and then squeezed through the filter into a region well. Membrane-bound antibody in well specific to A. actinomycetum comitans, P. gingivalis, and P. intermedia reacts with the plaque sample. Antigen and antibody complexes formed on membrane are detected by addition of an enzyme labeled second antibody together with a colored enzyme substrate. Separate dots will be formed and they indicate the presence of 3 different species. The advantages of this test, it can carry out simultaneous testing of multiple samples, has greater detection of bacterial colonization in deep pockets than shallow pockets. The disadvantages of this test is that multi-stage test and a subjective calorimetric endpoint. Omnigene test is a DNA probe system for a number of non-periodontal pathogens subgingival bacteria. It is applied genetic engineering to develop species-specific DNA probe tests for 8 periodontal pathogens. For example, P. gingivalis, P. intermedia, Echinella corrodens, Campylobacter rectus, Bacterius falciparum, and T. denticola. P. intermedia and P. gingivalis can be detected by the aid of purified DNA fragments, but A. actinomycetum comitans cannot be detected by this procedure. The advantages of this test is the reports provided in short periods of time and helps identification of number of known pathogens.
Now, I'm going to talk about PerioScan. PerioScan is a diagnostic test kit that utilizes BANA to assess a hydrolytic reaction. It detects the presence of bacterial trypsin-like proteases in the dental plaque. It can also be used for assessment of oral halitosis to detect the presence of 2 BANAS positive species on the tongue surface, which are Stomatococcus mucilaginous and Rhotia dentocariosa. It is also used to detect the presence of 3 periodontal pathogens in the subgingival plaque, which are Treponema denticola, Porphyromonas gingivalis, and Tannerella forsythia. These 3 microorganisms have trypsin that hydrolyzes BANA to give a positive reaction. So BANA test is actually a plastic strip to which 2 separate reagent matrices are attached. The lower matrix is impregnated with BANA. Subgingival plaque samples are applied to this lower matrix. The upper buff reagent matrix contains a chromogenic diazo reagent, which reacts with one of the hydrolytic products, which is B-naphthylamide of the enzyme reaction to form a blue color. The test is folded together so that the 2 matrices are coming in contact and is incubated for 5 minutes at 55 degrees Celsius. The blue color appears in the upper buffer matrix is permanent, and the intensity of the color determines whether it is a positive or weak reaction. So for the advantage of PerioScan, it is used to identify volatile sulfur compounds in halitosis patients. There are 4 disadvantages of this test, which is it has a lack of quantitative data. The specific bacterial agents that are responsible for enzyme production cannot be determined. Besides, they cannot identify the presence of other pathogens that do not produce trypsin-like enzyme. And finally, the results are qualitative and rely upon the operator's assessment at the colorimetric endpoint.
Here are the list of an example of the biochemical test kit that available in market and aid for diagnosis of the periodontal disease. We have PerioCheck, Prognostic, Pocket Wash, PerioGuard, Perio 2000, Topaz, Dipstick, Import of Nazri, and etc.
Alright, we start with PerioCheck. PerioCheck is the most rapid chest-side diagnostic for neutral proteases such as collagenases, proteinases, and elastases in GCF. So how do we do it? First, the GCF sample strip is placed on a gel containing insoluble dilabile collagen fibril and incubated. So in the presence of neutral proteases, the insoluble collagen dye complex is digested to release soluble dilabile fragment, which diffuse back into strip turning blue. With the development of gingivitis and periodontitis, the level of this enzyme in GCF started to be increased. Alright, next is prognostic. The system detects the presence of serine proteinase elastase in GCF sample. So how? The GCF is collected into the filter paper strip impregnated with a known amount of buffered elastase substrate labeled with a fluorescent indicator. Elastase on the test strip cleave the substrate during the reaction of about 4 to 6 minutes and release the indicator, and it is visible under fluorescent light. Elastase is accumulated at the sites of gingival inflammation.
Period 2000 system was designed to show the sulfate level digitally at each site. This is really usable as pathogenic microorganisms like Porphyromonas gingivalis, Porphyromonas intimedia would produce sulfates. Therefore, the volatile sulfate compound, by degradation of serum proteins like cysteine and methionine, can directly degrade the periodontal structures, aggravating periodontitis, which this will indicate the subgingival microbial load. Next is the pocket watch. Pocket watch was designed by a simple method of analyzing AST. It is used by analyzing AST at the chair site. It needs the presence of pyridoxal phosphate. AST will catalyze the transfer of an amino group of cysteine sulfuric acid by alpha ketoglutaric acid to yield the beta-sulfonyl pyruvate. The glutamate beta-sulfonyl pyruvate spontaneously and rapidly decomposes and releases inorganic sulfide. It reacts with malachite green, and this malachite green will convert itself from green color to colorless form. The colorless form will allow the pink dirhodamin B to show through. The malachite green is directly proportional to the AST concentration. Therefore, the pocket watch provides not only an index of cell death, but also the extent of the destructive pockets.
Lastly, Period biochemical test kit. So Period is based on the measurement of level of enzyme aspartate aminotransferase in the GCF samples. This commercial test consists of a tray with 2 test wells for each tooth and appropriate reagent for conducting the test. So this test also designed to be positive at more than 800 micro IU of AST activity and at negative value less than 800 micro IU. This test also indicated in the assistant or aid to diagnose in identification of active site at the time of the periodontal disease. It also to appreciate the result of the periodontal treatment. It also aid to monitor of high risk site.
Hi doctor, the next diagnostic test kit available in the market is the genetic test. The first genetic test is periodontitis susceptibility test, and it is for the severe periodontitis and commercially available. It evaluates the simultaneous occurrence of LL2 at the IL1 alpha plus 4845 and 1 beta plus 3945 loci. IL genetic susceptibility may not initiate or cause the disease, but rather may lead to earlier or more severe disease. The IL genetic test can be used to differentiate certain IL genotypes associated with varying inflammatory responses to identify individuals at risk for severe periodontal disease even before the age of 60.
So hello everyone, I will be explaining about another genetic diagnostic test kits for periodontal diseases, which is MyPerioID. So MyPerioID is actually a test that uses saliva to determine a patient's genetic susceptibility to periodontal diseases. So it actually assesses patients which are at higher risk of more serious periodontal infections, and this test actually requires the transportation of saliva samples to a laboratory for results. So how this MyPerioID test works is that it actually detects any genetic variations from the human DNA to check for polymorphism within the interleukin-1 gene. So as we all know, interleukin-1 is actually a major inflammatory mediator, and around 30 to 35% of the world population has this genetic variation in their DNA. So interleukin-1 positive individuals, they actually tend to have more aggressive and more severe infections. And MyPerioID test actually determines patients that are more susceptible to severe types of diseases, especially if the patients smoke. So this genetic variation among patients can actually increase their risk for severe disease or tooth loss by around 2 to 7 times when the disease is actually present.